So good evening, friends.
Good evening, sir.
Good evening, sir.
Okay, and this is the first biology test you people have written, correct?
Yes, sir.
Yes, sir.
Very good. How you felt the test, unit test?
Good, sir.
Good.
It's okay.
Any particular thing you found it hard? Anything you found it irrelevant?
No, sir.
Nothing. Okay, okay.
I found this learning the
only-
scientific names
Only
a
3
little.
chapters are there in this test. Therefore, it is quite easy for you. No problem. And the topics are quite big actually. And you can have one more test also, no problem. And, we are going to give you a lot of home assignment questions which are going to be quite relevant, and that may help you also. And whatever you feel hard, you can always ask us to clarify those doubts, and we are there to help you people, because we want to see that you are going to do well, as we are part of the team now, and our objective is to help you people to excel in your studies. And directly now, I'll come with the questions, and if you find any specific doubt, you can ask me. I'll try to clarify that. Okay. Now we move on to the first one. See, to the question paper, we start with 5 questions which are going to be 1 mark type. And remember, whenever we are going to ask you to answer a question for 1 mark, the answer is in single word or a single statement, depending upon the kind of question that is going to be asked. And here, mention the function of nephridia. The moment we talk about nephridia, we know we have talked about excretory structure in animal kingdom. And later in human physiology also, we are going to talk about excretion and elimination of metabolic waste, where we have a list of excretory structures that start from protonephridia. What we are going to see in case of flatworms, platyhelminthes, I hope all of you remember. And protonephridia are also called flame cells. And then comes the next level of structure, that is nephridia, what we see in case of annelids. The moment I say annelid, I hope all of you remember earthworm, nereis, leech, all this. And
Yes.
excretory structures are basically meant to eliminate the metabolic waste. And the metabolic waste is going to be in the form of urine. And metabolic waste, when you talk about, it may be nitrogenous waste products or respiratory waste gas like carbon dioxide, and it can be anything that is found in excess. And when we talk about nitrogenous waste product, we mention ammonia, urea, uric acid. And when you come to human physiology, I'll give you the details, nothing to worry. And nephridia, we are going to see in case of annelids. Earthworm type study is also there in structural organization in animals. If you remember, where we come across 3 kinds of nephridia. Segmental nephridia, which is also referred to as septal nephridia, integumentary nephridia, and pharyngeal nephridia. Integument, have you heard of this word integument? Emmanuel?
Yes, sir.
What do you mean by integumentary?
I'm not sure what it means. I've
Okay, no
heard
problem.
it before.
Shamita, you know it?
No, sir.
All of you record it. Integument means skin. Integument, skin. Skin is the largest organ in the body, correct? If you consider skin as an organ, it is the largest organ in the body. And these are the nephridia which open directly, okay, to outside. So they are opening into the skin, thereby integumentary, like our sweat glands. Whereas pharyngeal and septal nephridia open into the digestive tract. And if we talk about the function, two functions you should know, which is given here. One is osmoregulation and other one is excretion. Osmoregulation comes from the word osmosis. Osmosis relate to water and solutes, correct?
Yes, sir.
Yes, sir.
Very good. Net movement of water molecules from the region of
High water potential to a
higher
region
water potential. Very good, very
of
good.
low water potential.
To the region of lower water potential. Excellent. And how much of body fluid should be there in our body? It has to be taken care of. Our kidney also perform the same function. Excretion relates to elimination of metabolic waste. Mention the function of every day. If you write any one of the two, you will get the marks. Because functions they have not asked. Here function, there will be just one word. Osmoregulation, if you write, you have to get a mark. If you write excretion, you'll get a mark. Is it clear?
Yes, sir.
Very good.
Yes, sir.
So go back to some. Osmoregulation, look, maintenance of solute concentration and water in the body. How much of solute I should have, how much of water I should have in my body. Our kidneys are wonderful osmoregulatory structures. I give you one reason, simple example. During summer, volume of urine output is less. During the winter, because of the cold, volume of urine output is more. What may be the reason? The reason is simple. During summer, we are going to sweat. Thereby, we are losing water by other means. So thereby,
Yes, sir.
urine output is less. During winter, our sweating is not there. So urine volume is going to be more. I hope all of you got it.
Yes, sir.
Good. Elimination of metabolic waste from the body is excretion. Metabolic waste means in the cell functions happen, chemical events, what we call as metabolism, and the waste obtained or derived there is eliminated. Okay, we come to the next slide now. See, I told you that earthworm is an annelid. In annelids, we have nephridia. This is just a slide showing earthworm and how the nephridia are arranged here. You can see them. Nephridia labeled in the gut, they are opening. So these are septal nephridia, what is shown here. Okay. Now we can move to the next slide. The second question is, what is hypogynous flower? I hope all of you know. When you talk about flower, flower is a reproductive structure. Okay. Flower is a reproductive organ. Typical bisexual flower has 4 whorls. From outside to inside, they are calyx, corolla, androecium, and gynecium. Am I right?
Yes, sir.
Very good. You remember it. And the unit of calyx is called as sepal. The unit of corolla is called petals. Androecium is a male reproductive organ, and it is made up of stamens. That is the unit. And gynecium is a female reproductive organ, and the unit is pistil or carpel. Correct? On the basis of position of gynecium compared to the other whorls, we are going to classify the flower into 3 categories. They are hypogynous, perigynous, and epigynous. And here the question is hypogynous. Beneath the gynecium, other whorls are located. That means other whorls are going to be inferior in position. Gynecium is superior. So superior ovary we mentioned. I hope all of you got it.
Yes,
Look
sir.
here.
Yes, sir.
Gynecium occupies the highest position, that is superior, while the other part situated below it, inferior. And the ovary in such flower is said to be superior because it is the highest position. And 3 examples are given in your book. I want you to remember the example. They may ask you What is hypogenous flower? Give example. Give two example like this. So mustard, China rose, brinjal. These are the examples I want you to remember. At the same time, if I tell you about perigynous, I hope all of you know perigynous is half inferior. So look here, the gynecium is going to be surrounded by the other folds. There are two diagrams are given. It is in the center. Epigynous means the other folds are at a superior position. Gynecium at inferior position. Is it okay?
Yes,
Yes,
sir.
sir.
Very good. We can move to the next question. What is parthenocarpic fruit? Normally, when we talk about fruit, we know it is a feature that we are going to see in flowering plants. Because in flowering plants, the seed is covered by seed coat. And the ovary develops into fruit, ovule develops into seed. And many times, the fertilization occurs, okay, and as a result, seed develops. In rare cases, the fruit develops without the fertilization. And we call such fruits which are developed from unfertilized ovary as parthenocarpic. Remember, fruit developed from ovary, seed developed from ovule. These are the two types. And here there is no fertilization and they are seedless varieties. That's the reason you, in the slide, just I have given you a bunch of grapes. Is it clear? So fruit formed from unfertilized ovary are called parthenocarpic. Are called parthenocarpic. And in plant physiology, last chapter, you study about growth regulators. And one of the growth regulator mentioned is gibberellins. Gibberellins are going to be used to get seedless varieties. They have given the example of guava, seedless. Is it clear? And of course, we have even seedless tomato. Okay, grapes are quite popular. Thereby, I have given that. So, which growth regulator is usually used to induce parthenocarpic development? The answer is gibberellins. Of course, one more growth regulator also is used called auxins. That is not mentioned in the text. Thereby, I have not quoted it. Okay. And commercial significances, these are seedless varieties, and we prefer seedless varieties when it comes to the fruits. Grapes, you take seeded, you get seedless. Commercially, we prefer seedless. Agree?
Yes, sir.
Shall I move to the next question now, friends?
Yes, sir.
Okay, we go to the next slide. Define mesophil tissue. We have studied plant tissues. We broadly classify plant tissue into meristematic tissue and permanent tissue. When we come to permanent tissue, we mentioned that they are made up of homogeneous or heterogeneous mass of cells with specialized functions. When it comes to homogeneous mass, we are going to mention three types of simple permanent tissues: parenchyma, collenchyma, and sclerenchyma. And parenchyma is going to be found in soft parts of the plant, and parenchyma that is found beneath the upper and lower epidermis of the leaf is called mesophil. So what is mesophil? Parenchymal tissue present between upper and lower epidermis of the leaf. And here I want to give you a little more. The leaf is a specialized organ for photosynthesis, correct? And thereby the parenchyma which represent the mesophil carry large amount of chloroplast. And in case of dicot leaf, we find dots, which is also called dorsiventral leaf. We find two types of parenchyma tissue representing mesophil. Given there, palisade and spongy parenchyma. Here it is labeled as palisade mesophil and spongy mesophil. Spongy means loosely arranged like a sponge with lot of intercellular spaces. And remember, upper and lower epidermis are also called dorsal epidermis and ventral epidermis on the basis of position. They are technically called adaxial epidermis, abaxial epidermis. Is it clear? For you, the answer is very simple. Parenchyma containing chloroplast present between upper and lower epidermis of the leaf. If you say dorsal and ventral epidermis, fine. If you say adaxial and abaxial epidermis, fine. So different terms we can use. So here, just to show you the parenchyma with green color, you can see that is the mesophil. I hope you have got it. Mesophil is the parenchyma present between upper and lower epidermis of the leaf. Important feature is contain chloroplast and specialized for photosynthesis. Agree? Have you understood? Okay, we move to the fifth question. Where do you find bulliform cells or motor cells? This is very important even in plant physiology. So here I have shown a section of the leaf where in the upper epidermis you can see the labeling bulliform cells. Bulliform cells are also called motor cells. They are thin-walled, colorless, and enlarged cells compared to other epidermal cells. Now I want to tell you one thing. All of you remember, epidermis cells themselves are colorless. Then if you ask me, sir, why the leaf appear green in color? The reason is because of the mesophil containing the chloroplast. Once the leaf become old, the chloroplast get disintegrated and other pigments show up, like yellow color. I hope all of you got it.
Yes.
Green in color.
Okay. Now carefully come to one more thing. The monocot leaves are going to have bulliform cells, not dicot. Monocot leaves are called isobilateral leaves. Dicot leaves are called dorsiventral. And in monocots, stomata is present both in upper and lower epidermis. We call that condition as amphistomatic condition. Because of the presence of stomata on both the sides, monocots are the one which lose large amount of water. And thereby, there is a need for a mechanism to cause rolling and unrolling of leaves. Motor cells play an important role in rolling and unrolling to regulate the water loss, to minimize the water loss. And when water is less present, motor cells become flaccid, causing rolling, decreasing the surface area of exposure, and less water is lost. When water is more, they become turgid, and as a result, they can lose water. So this is very important. We ask you, what is the significance of bulliform cells in isobilateral leaf or monocot leaf? Then you can answer. Now look here, what is the answer given? Large, empty, colorless cells present in adaxial or dorsal or upper epidermis, any one of the term you can use. Of isobilateral leaf. Isobilateral means it is going to be uniform in the arrangement of the tissue, both on the dorsal side and ventral side. I told you, amphistomatic, stomata are present on both sides. Mesophil, look there, is not differentiated into palisade or spongy parenchyma. It is undifferentiated parenchyma, of course, containing chloroplast. The only thing is on the dorsal surface, that is adaxial epidermis, some cells are modified to become motor cells because the dorsal surface is the one which is exposed to the sunlight. And they are the one which have to regulate how much of water they have to use. Shall I come to the next question now?
Yes, sir.
Yes.
Okay. See, now we move to the short questions, and here each question is going to carry 2 marks. So first thing, differentiate between chordates and non-chordates. All invertebrate phyla which do not have notochord or pharyngeal gill clefts, is it clear? And dorsal tubular nerve cord, they are called non-chordates. Non-chordates are also called invertebrates. And all phylum except phylum chordata come under non-chordata. Am I right?
Yes, sir.
Very good, very good. From phylum Porifera, represented by sponges, to phylum Hemichordata, they are non-chordates. So here, differentiate between chordate and non-chordate. How many differences we have to write? And what is the scope in terms of differences? Then remember, since it is not mentioned, we are going to write 4 differences. Minimum marks is going to be half a mark, and thereby half into 4 is 2 marks. This is the way we are going to give you this skill. Sometimes we may ask you, give any 2 differences. Then we look for only 2 differences. If you write more, no problem. If you write more, if any of the difference is wrong, we ignore it. We give benefit of doubt to the student. So correct one, we are going to look into it. Okay?
Yes, sir.
Look, notochord. Notochord is going to be present in chordates, absent in non-chordates. Please remember, there are 3 fundamental characters of chordates. Presence of dorsal hollow notochord, dorsal solid notochord, dorsal hollow nerve cord, and pharyngeal gill clefts. They must be present at least sometime in the life of an organism. Not necessarily they have to be present throughout. None of us have notochord. We have vertebral column. Notochord is replaced in us. Sir, we don't have notochord. Are we not chordates? Then remember, in our embryonic life, we had the notochord. We had pharyngeal gill clefts. We don't have gill clefts now. I hope all of you got it.
Yes, sir.
Now, we
Yes.
may ask you to list out the 3 fundamental or basic characters of the chordate. Then you have to write that also. So first, notochord, present in chordates, absent in non-chordates. Second, central neural system, nervous system. And here I am talking about nerve cord, dorsal, hollow, and single. Whereas when it comes to non-chordates like annelids, arthropods, we are going to find the nerve cord which is ventral in position. And it is not hollow, it is solid. And there are a pair which are going to run together, thereby ventral, solid, and double. And the third difference, pharyngeal gill clefts. So pharynx is perforated with gill slits. Gill slit is also called gill cleft, C-L-E-F-T. And gill slits are absent in the non-chordates. Please remember, non-chordates can have gills. Mollusca, we have gills called Ctenidia. In marine annelids, we have the gills. Is it clear? But here we are talking about gill slit openings. And heart position. This is outside the 3 fundamental characters. First 3 I have touched the fundamental character, the position of the heart. In case of chordates, in case of human, example for chordate, the heart is ventral in position. When it comes to non-chordate, it is dorsal in position. And last thing, post-anal tail. Normally, in invertebrates, anus is found in the terminal part of the body, posterior most part of the body, except nematodes. Judith, have you heard of nematodes? Shamita, heard of nematodes? Roundworms, Ascaris lumbricoides.
Yes, sir.
Very good. There,
Second.
the anus is found, okay, little earlier to the terminal part. Except nematodes, in all other where anus is going to be present, it is terminal part. Only phylum where post-anal tail is found among non-chordate is Aschelminthes, Nemathelminthes, also called Nematoda. Write a note. So in non-chordate, post-anal tail is absent. Exception is Aschelminthes. 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 I have given you one exception. Okay. This is for your CET and NEET. I want you to remember it. So here I have mentioned 5 major differences. And if you write any of the 4, you get half mark each. Thereby easily you can score your 4 marks. We come to the next one. Next lecture. Assign the following animals to their respective phyla. This is the normal kind of a question or we are going to ask you in the exams. Thereby it is a very relevant question. So jellyfish, they have given the common name here. Jellyfish, sea star. I hope all of you know jellyfish is Aurelia. Aurelia is commonly called jellyfish because of the translucent body. And it is marine. And it belongs to phylum Cnidaria Coelenterata. Just if you say Cnidaria or Coelenterata, any of the 2 terms, you get 1 mark. That is the scope here. Sea star, starfish, Asterias. I hope all of you know it. And the moment you write Echinodermata, you get a mark. Easy. I hope all of you got it.
Yes, sir.
Yes, sir.
Okay. We move to the next question. What is venation? Explain the types. See, I told you about the monocot leaf is called isobilateral leaf. In the same way, dicot leaf is called dorsiventral leaf. You have studied plant anatomy, thereby you know TS of isobilateral leaf, TS of dorsiventral leaf. And when you look into the veins and veinlets, veins or veinlets are nothing but strands of vascular tissue, that is xylem and phloem. And nature of distribution in monocot and dicot is going to be different. In monocots or isobilateral leaf, the veins are parallelly distributed. In case of dicot, it is in the form of a network. We have a midrib vein and its branches. We call it as reticulate venation. So, question is, what is venation? So, normally we go for 1 mark for the definition, but here, since 2 types you have to explain, 1.5 mark out of the 2 is kept for the explanation of the type. Definition we have kept half a mark. Otherwise, may ask you, what is venation? Mention the types. Venation, then we give you 1 mark. Mentioning the type, half mark each. You know. And here, look, arrangement of veins and veinlets in the lamina of the leaf lamina is called venation. What are the 2 kinds? Reticulate and parallel. Reticulate means network like. Veinlets form a network as in leaves of dicot plants, that is dorsiventral. Any of the dicot example you can give, China rose and people we have given. Parallel venation is a feature of monocots. Veins are parallel to each other as in leaves of monocot plants, grass, maize, sugarcane, all this. I hope all of you got it.
Yes, sir.
Oh,
Yes, sir.
shall I go to next question now?
Yes, sir.
Next slide, please. Here is a slide to show you the nature of venation. In a dicot plant, you find reticulate venation. There are exceptions. Parallel venation also you can see in grasses you are going to find them. Monocots you are going to find them. We move to the next slide. Write the differences between open vascular bundle and closed vascular bundle. See here, we have asked you to give the difference. And thereby, if you write 2 differences, enough. Or if you define what is open vascular bundle, what is closed vascular bundle, and give an example, we give you a mark. So 1 mark each. Now, look here. Open vascular bundle, closed vascular bundle. When you talk about vascular bundle, if xylem and phloem occur together, we call it as conjoined. In conjoined vascular bundle, we are going to have collateral and bicollateral vascular bundles. When you come to collateral, we look into open and closed type. So in your book, they have not looked into collateral. They have talked about conjoined only, and then they have talked about open and closed. If cambium is present between the xylem and phloem, we call it as open vascular bundle. It is open for secondary growth. Open secondary growth means it is going to contribute to secondary xylem and secondary closed. Closed vascular bundle means cambium is absent between xylem and phloem. Thereby, there is no secondary growth. And remember, secondary growth is a feature of dicot stem and root. And secondary growth in dicot stem is explained in your book. In monocots, there is no secondary growth because there is no cambium. Is it clear?
Yes, sir.
Now, what are the differences we have given here? Cambium present between xylem and phloem, simple. Cambium absent. You can say conjoined vascular bundle, where cambium is present between xylem and phloem, we give you mark second. Second, possess the ability to form secondary xylem and phloem due to the presence of cambium. Do not possess the ability to form second. They may ask you, why in monocot secondary growth is absent? The answer is, they have a closed vascular bundle where cambium is absent. Simple. Different style, we can ask you this question. Example given, present in dicot stem, present in monocot stem. And dicot root also shows secondary growth, where cambium is going to be produced from simple permanent tissue. Okay? So here we are talking about the stem. Because in the roots we find radial type of vascular bundle. I hope all of you know it. Next question.
Yes, sir.
Next slide we come to. Mention the functions of scleroderm. We already know there are three simple permanent tissues: parenchyma, collenchyma, sclerenchyma. Out of the three, parenchyma and collenchyma are simple permanent living tissue, whereas sclerenchyma is a dead tissue at maturity. Sclerenchyma has secondary wall deposition in the form of lignin. Wherever you have secondary wall deposition in the form of lignin, they provide one function, that is mechanical support. They are part of woody tissue. Okay. So here we have asked you to write the function. Since only one function is there, we expect you to tell what is sclerenchyma and then function. So first statement is, what is sclerenchyma? Simple permanent tissue formed of dead cells with thick and lignified wall. Lignified means lignin deposited. And that is going to signify the function itself. What is the function? Provide mechanical support to the plant artery. So scheme, what is sclerenchyma? 1 mark. Function 1 mark. Got it? Okay, we come to the next one. See, we have not asked about commercial significance of sclerenchyma. Okay, where we talk about using the sclerenchyma fibers as jute, flax. I hope hemp, you have heard of all this. Coir. Okay, coir is used for rope making also.
Yes,
Okay.
sir.
Next slide, please. We come to short questions where each question is going to be for 3 marks. So you are expected to write in there. Describe the structure of parenchyma with a label diagram. Here we are asking to describe the structure and a diagram. So remember, in biology, diagram is going to be given equal weightage to describing. If it is a 3 mark question, diagram itself is going to be 1.5 marks. Have you got it? And label diagram is going to, you get a marks. If it is unlabeled, 0 marks. You might have beautifully described it. We will not give you full marks. We give you 1.5 marks only, because scheme says diagram is required and labeled diagram. If it is not labeled, you don't get a mark for it. And if it is 1.5 marks, minimum 3 important major labeling must be there. Here, how many labeling are there?
5, sir.
5 labeling. Minimum how many are required? 3. That
3.
doesn't mean don't be very calculated. You can give the labeling what are you think important. So, but when I value the paper, I look for 3 labeling, half a mark for each correct labeling with the diagram. Okay. So here We have given you parenchyma. Parenchyma, as I told you, is a simple permanent living tissue. And remember, as a living tissue, it is going to have cytoplasm, which is granular, and a nucleus, and they are going to show a vacuole. And cell wall is going to be very thin, made up of only cellulose. Secondary wall depositions are absent. In your book, they say thin cellulosic cell wall, finished, of a mark. If you write 3 important points to describe the structure, you get half mark. We go to the next slide. Look here. Parenchyma is a simple permanent living tissue. Cells are generally isodiametric, means equally expandable in all directions. They may be spherical in shape, they may be oval, they may be round, they may be elongated. Sir, if it is isodiametric, how can it be elongated? Then remember, it is a living tissue made up of thin wall. When other tissues are there, they can put a pressure, and as a result, the cell becomes elongated. Have you understood what I said? Palisade parenchyma, mesophil of leaf. That is elongated cell. Cell wall is thin and made up of cellulose. Second point. Cells are loosely arranged with intercellular spaces. This is one common feature. But when other tissues are added, they may become compact and intercellular space may not be seen. Your book has given that. So thereby, we have mentioned here in this statement, cells are loosely arranged with intercellular spaces or tightly packed without intercellular spaces. If somebody asks you, so normally parenchyma, intercellular spaces are present, but you say here without intercellular spaces, then you have to tell because of the presence of the other tissues that exert a pressure, they may be packed. And as they are packed, you may not find the intercellular space. And function we have mentioned, even if you don't write the function, no problem. If they ask you to state the function, definitely you have to write. And remember, if it contains chloroplast, it's of photosynthesis. And we call such a parenchyma as chlorenchyma. I hope all of you know. What is chlorenchyma? A specialized parenchyma containing chloroplast help in photosynthesis is chlorenchyma. Storage. Basically, it is a storage tissue. In the pith, we find parenchyma where cells are compactly arranged because of the other tissues, and it is going to store. It may store water, it may store food in the form of starch. They may be secretory. We get resins, gums, tannins, aromatic compounds, essential oils. All these are going to be products of or produce of the parenchyma. In addition, we have a specialized parenchyma you have read called Aerenchyma. Have you heard of it, Shamita? Aerenchyma, A-E-R-E-N-C-H-Y-M.
Yes, sir.
Aerenchyma
Yes,
has
sir.
prominent intercellular spaces present in hydrophytes, that is aquatic plants, and it provides buoyancy. Hydrilla, classical example.
Center float.
Okay, aquatic. Shall I go to the next question now?
Yes, sir.
Emmanuel?
Yes, sir.
Okay, next question. Next slide. Write the differences between hardwood and sapwood. So I hope all of you know, we have studied this in structural organization in plants, plant anatomy, secondary growth. Hardwood, sapwood. I have given in the bracket the other names, technical name. Hardwood is called duramen, sapwood is alburnum. Here you have to write the differences, and if you, since it is for 3 marks, you write 3 differences, 3 marks. Hardwood is present in the core of the plant organ stem. That means in the center or innermost region of the plant, heart, center. Sapwood is found at the periphery. Hardwood is going to be dark colored, because of which, as it is going to be long lasting, cannot be damaged by microbes or insects. It is used commercially important. It is used in making of furniture, doors, windows. I hope all of you know. Sapwood is the outermost, which still serve the functions of ascent of sap or inorganic transportation, water and mineral salts. So look here, what are the differences we have given? Hardwood is central or innermost region of stem. Sapwood is peripheral region of stem, present in the peripheral region. Dark brown in color due to deposition of organic compounds like tannins, resins, gums, aromatic compounds, essential oils. I told you, parenchyma is secretory. What happens means when hardwood develops, the parenchyma is going to be crushed, and through the pits of the xylem, it enters and blocks the lumen, and the secretions get deposited, and they also stem. We call them as extractive. That word extractive is not used in the book. They have mentioned this. I want you to remember what is that which gives the color to the duramen or hardwood. And such extractives will not be there. They'll be light in color. And since they are going to be seasoned because of the resins, gums, tannins, they become resistant and durable, duramen, to attack of microorganisms and insects. Whereas sapwood is vulnerable to attack of microbes. I cannot use sapwood for furniture. If I use sapwood for furniture, it is not long lasting. And last one, hardwood has lost the function of ascent of sap or inorganic transportation because it is blocked, not involved in conduction of water, but it is very strong. So it gives mechanical support to stem. Whereas sapwood, sap means ascent of sap. It is conducting the sap. Okay, water and inorganic nutrients. So that is mentioned. Minerals. So any three differences, three, we have given it four. We go to the next slide. What is phyllotaxy? Explain the type with example. It is one of the easy questions according to me. So this is for 3 marks. Phyllotaxy, half a mark or 1 mark. If they ask you to mention only the types, then 2 marks for mentioning the type, 1 mark for the definition. Here you are asked to explain with example. This can be asked for 5 marks also, remember. So if for 5 marks, if this you write this much, you will get your score. Phyllotaxy relates to arrangement of leaves on the stem or branch of a tree or a plant. And when you talk about phyllotaxy, there are 3 types of phyllotaxy. I hope all of you know. Alternate, opposite, and whorled. Leaf are borne on the point of a stem or a branch called node and internodes. So look here, alternate, single leaf at a node in alternate manner. China rose, hibiscus, mustard, brassica family. Opposite, if the 2 leaves are going to be on opposite side at the internode part, at the node, then it is called opposite. We find it in Calotropis, in guava. Whorled, if you find more than 2 leaves at an internode emerging from a internode point, you call it as whorled. More than 2 leaves in a whorl at a node. Example, nerium and alstom. So scheme, phyllotaxy definition, half a mark. Types, 1 mark. Explanation with example, half mark each. So if you mention all the types, 1 mark. Definition, half mark. So 1.5 mark. Explanation for each one. What is alternate? What is opposite? What is whorl? Half, 1.5. So 3 marks. Have you got it?
Yes, sir.
Okay. We go to the
Yes,
next
sir.
question. See, this is a diagram just to make you remember. In alternate, you find alternately the leaves are going to emerge from the node region. Whereas in opposite, 2 leaves at the node. In whorl, more than 2 leaves. Here they have shown you 5 leaves. Okay.
Yes,
Next
sir.
one. Next slide. Next. Okay. List out any three salient features of echinodermata. Beautiful question. Please remember, salient feature means diagnostic characters. They are characters which are unique to that phylum. They are not general characters. General character means any character. Salient feature means unique character or diagnostic feature. Is it clear? So normally when we talk about unique character, one unique character, 1 point, 1 marks. So here three, very clear for 3 marks. Echinodermata, the moment we say, we call them as spiny skinned animals, correct? Spiny skinned invertebrates. So spiny bodied organisms with endoskeleton of calcareous ossicles. Please remember, they are having spiny skin. The endoskeleton is there, not exoskeleton. Ossicles are what they may ask you in the CET NEET. Then I want you to remember endoskeleton. Second, 100% these are marine organisms. When I say marine, coelentrates, majority of marines. Ctenophores, 100% marine. So there are groups which are marine. Hemichordate, marine. Cephalochordate, marine. Urochordate, marine. So along with that, you have to write triploblastic with organ grid of organ system level of organization. Adults, very, very important unique feature. These are triploblastic organisms, but they exhibit radial symmetry as an adult organism. Larvae are bilaterally symmetrical. Larvae is more evolved compared to adult. And another important feature is water vascular system. Unique feature. Nowhere else you find this. Nowhere else you find adult showing radial symmetry, larva showing bilateral symmetry. And excretory system is absent, very important. Because water vascular system itself has taken part of elimination of metabolic waste. It has taken the role of gathering the food and involving it. Of course, they have mouth, they help in picking the food and passing it. Respiration, locomotion. We go to the next question now, sir.
Yes, sir.
Write the general characters of phylum Annelida. Look, there we have asked you the salient feature. Here it is general characters. When it comes to general characters, if it is going to be 3 mark question, it is 3 question, 3 or 6 we ask you. If it is 5, then you can write 5 or even 10. We mentioned, if it is 5, we mentioned. So here, any character can be written. It is not salient feature. Triploblastic, bilateral symmetrical, coelomate, metamerically segmented is unique character. Of course, we find metamerism in 3 phylums: phylum Annelida, Arthropoda, and Chordata. In Annelida, we find metamerism both externally and internally. That is unique. Whereas in arthropods, cockroach, external metamerism is there. In chordates, metamerism only in the developmental stage or embryonic stage. Some organs may show sign of metamerism, segmentation we call it. And general character, there may be aquatic or terrestrial, free living or parasitic. Body surface shows that metameres, segments, and important, longitudinal and circular muscles help in locomotion. First time the muscle make their appearance in Askelmathis, where they are longitudinal muscles. Whereas in annelids, we also find circular muscles. And in case of earthworm, we have locomotory structure called setae or chetae. In nereis, we are going to find parapodia, which is also going to help in respiration. Digestive system is complete, very important, because in flatworms, digestive system appears for the first time. It has only one opening mouth. We call it as incomplete. Whereas here, it has mouth and anus at extreme ends. We call such a digestive system as complete. General characteristics. We go to the next slide. Sir. Respiration. See, in earthworm, classical example, skin help in respiration, which is covered by cuticle. We call it cutaneous respiration. Cuticle is the respiratory structure. There are aquatic organisms where we have the gills. We call it as bronchial respiration. Nereis, classical example. Of course, I can give you examples like Chitopterus, Aranicola, which you don't study. So, no need to worry about it. Nephridia, in the first question we talked about it, osmoregulation and excretion. Nervous system, I told you, in case of annelids, arthropods, mollusks, we are going to find double ventral solid nerve cord. And here, nerve cord will be there, ganglia will be there, from the ganglia, nerve connections are there. So, consists of paired ganglia connected by lateral nerves to double ventral nerve cord. If you say double ventral nerve cord, ganglia and nerves, you get your marks. And we find unisexual and bisexual forms. Unisexual means separate male and female, Nereis. Bisexual, earthworm, leech. Leech is a parasite, remember. Reproduction sexual, any character. So what is mentioned here? Mention any 6 general character, half mark each. So 3 marks you get it. It is better when you in the test you write more. Suppose we decide only 3 characters are enough. We consider 3 correct characters for it. Is it clear? If they mention only 3, you are very clear, you can write them. If they mention 6, you write 6. No need to write more than that. Here we have given you more because you can remember it. We
Yes.
come to the next question. Now we come to long question. Okay. Only one question you have to answer for 5 marks. Where internal choice is there, out of the 2, you can select one. Write any 4 salient features of phylum Arthropoda and name any 2 economically important insects. Classical question it is. So here it is very clear. 4 salient features and 2 economically important insects. So what you write, you are writing 4. Each salient feature 1 mark per you. And 2 insect names, half a mark each. So 4 plus 1, 5. Very clear. See, scheme I am giving you. Scheme, if you understand, when you write the answer, you present an answer, you can always be very clear. Look here. Chitinous exoskeleton is a salient feature. So body is covered by chitinous exoskeleton. Body division. We may divide the body into head, thorax, abdomen. In case of prawns, head and thorax are fused, cephalothorax and abdomen. We also have the term like prosoma, mesosoma, metosoma, not given in the book, don't worry. Then arthropoda, jointed appendages, characteristic, unique. Respiratory structures. Remember, arthropods are represented by highest number of species on earth. And there are 4 different types of respiratory structures. Thereby, you can read. They have respiratory structure like gills, book gills in aquatic forms, book lungs, trachea. When you talk about gills, prawn is a classical example. When you talk about book gills, king crab, limulus is an example. Book lungs, scorpions, spiders. Trachea, cockroach, insects. Then excretory structures, again, we have Malpighian tubules in terrestrial insects, coxal glands in spiders and scorpions, antennal gland in your human physiology, introductory aspect, they have mentioned it, and prawn, you should always remember, prawns, lobsters, they are also called green gland. Circulatory system, important, open tight, very few phylum are going to show. The first phylum to show clear-cut circulatory system is Arthropoda. Sense organs. When we talk about sense organs, here we are going to find eyes, simple or compound eyes. The only phylum where many members are going to show compound eye is Arthropoda. Cockroach, prawn, we can show. And there are sensory structures called antennae, antennules, anal cerci, statocyst, balancing organ. Next slide. Economically important insects. So they have given you 3. You are asked to write 2. You write any of the 2. Apis, honeybee, Apis mellifera, Apis dorsata, Apis florae. In practicals you study, thereby you should know it. Bombyx murray, silk moth. And silkworm is the larvae of the silk moth. Lacifer, black insects. These are economically important insects. So what is the scheme? Any 4 salient features, 1 mark each. 2 insect name, half mark, 5 marks. Now we come to the last question. What is estivation? Explain the type with example. See, actually, there are 5 types of estivation. In your practicals, they have mentioned all the 5 types. In theory, they have mentioned 4 types. So you can write all the 5, no problem, because this is for 5 marks. Since here the definition is what is estivation, 1 mark for definition, then as 4 types are mentioned in the book, you write the 4 types, explain. 1 plus 4, 5. So what are the 4 types? Valvate, estivation, twisted, imbricate, vexillary. Estivation refers to arrangement of petals and sepals in a bud stage. In the beginning, when I talked about hypogynous flower, I said a typical bisexual flower has 4 organs: calyx, corolla, androecium, and gynoecium. And I told you, calyx is made up of unit called sepal, corolla is made up of unit called petals. In a bud stage, how they are arranged, that speaks about estivation. Look there, the definition is given. The mode of arrangement of sepals or petals in floral bud is called estivation. Means in a bud, flower bud. When you look into valvate, non-overlapping type. I hope all of you can see non-overlapping type. In case of twisted, all are overlapping in a bud stage. Valvate we are going to find in calotropis. Twisted we are going to find in China rose. Imbricate. One is completely in, one is completely out, 3 are in and out. Imbricate. One calyx, that is sepal or petal, is completely in. One is completely out. Other are 3 in and out. Whereas in imbricate, which is in twisted, they are all in and out. And imbricate, when you talk about it, we are going to find in gulmohar, cassia. Vexillary, very, very important. Vexillary estivation. Pepilinaceous corolla. 3 families you have studied in plant morphology. 2 dicot families, 1 monocot. Dicot family, if I ask you, anybody tell me what are the 3 families you have studied?
Solanaceae, Liliaceae, and
Solanaceae, wonderful. Liliaceae. One more. Fabaceae. What are the 3? Fabaceae, Solanaceae, Liliaceae. Fabaceae and Solanaceae are dicots.
Yes.
Liliaceae is monocot. And
So,
vexillary estivation is a characteristic feature of Fabaceae. And it is called pepilinaceous. And here you are going to see one very large, okay, petal, which is standard petal. That is posterior part of a flower. And then you are going to find 2 which are also called wing petals, also called lateral petals. The other 2 are fused, that are going to represent the anterior ring. They are called keel, K-E-E-L-Q. Also called pepilinaceous corolla. Vexillary estivation is a characteristic feature of pepilinaceous Fabaceae family, P. You can give any of the Fabaceae example. Bean, you can give the example. So here 4 marks for 4 and 1 mark for the definition. We go to the next slide.
So
It
got
types
it.
of estivation. Valve. Sepals or petals just touch one another at the margin. If you say without, okay, overlapping, they are continuous. Calotropis, I told you that. Twisted. Sepals or petals overlap the next sepal or petal. I have given example like China rose, cotton, lady's finger. All the three examples are given in the book. Thereby we have mentioned. Imbricate. Margins of the sepal or petal overlap one another, but not in any definite direction. One completely out, one is completely in, other in and out. Quincuncial, there is one more that is there in the practical. Two completely out, two completely in, one is in and out. Imbricate, one out, one in, three in and out. Last, maxillary. Largest petal overlaps the two lateral petals, which in turn overlap the two smallest anterior petals, which we call wing petals. And heel. Example, Fabaceae family members, all Fabaceae family members. Just two examples are mentioned here, bean and heel. Skin. Definition 1 mark, type, explanation with example, 1 mark each. 4 types you are expected to mention. Each, what is it? What it means? What is the example? 1 mark. So 4 plus 1, 5 marks. So thereby I have finished. I come to the last one. Next slide. Just to thank you people for your patient hearing. I hope you understood. If you have any doubt, you can always ask us to clarify. We are there to clarify your doubts.
Sir, I've got one doubt.
Okay, I have taken 1 hour. I don't want to get more. Okay, God bless you people.
Sir, I've got one doubt.
Please.
So in imbricate aestivation, is it always that one is inside?
Yeah.
inside,
You go to the practical manual.
Yes.
Okay. In the practical manual, you are going to find 5 types. And here they have not mentioned because they have not mentioned quincuncial. Imbricate they have taken for both imbricate as well as quincuncial. Okay.
Okay. Yes, sir.
You remember always that.
Yes, sir.
Okay. You can write down out of 5 petals, 1 is completely internal.
Yes, sir.
1 is completely external.
Yes, sir. And the other three are in.
Other 3 are in and out.
Yes, sir.
Quincuncial, Quuncial. Here 2 in and out, 2 completely in, 2 completely out, and 1 in and out.
Yes, sir.
Okay, thank you. Good night. Thank you, Kishan.
Thank you very much, sir. All the best, students. Thank you.
Thank you, sir.
Thank you, sir.
You are.
Good night, sir.
Thank you, sir.
Good night. Bye.
Thank you, sir.
See you, ma'am. Good night.
So do we have class tomorrow? CET class tomorrow?
No, Iman.
No, okay. Just confirming.
Sure.
Because if not, I have to bring my folder. So
Sure.
Thank you, sir. Bye, sir.
Thank you, Emmanuel. Bye. Bye, Kelsey.